Okay, on lab 5, you will do a hands-on exercise on DNA's agarose gel electrophoresis. So I know most of you have done this before, so this will be an easy lab hopefully. So we talked about the theory of this technique. So agarose gel electrophoresis is basically separation technique, as we already said in the lecture. So it'll separate out nucleic acid molecules based on their sizes, right? And when you do this experiment, there are some things for you to consider, of course. Especially you have to prepare your own gel. When you do that, then how do you make a gel? So what's the concentration of agarose gel you need to make? So we talked about the concentration being between about 0.8 to 1.5%. So depends on the size of the DNA molecules you want to separate out. You can make a lower percentage gel or higher percentage gel. The lower the percentage of the gel, you will have larger size pores in the gel, so it'll be easier for you to separate out large size DNA molecules that have like 10 kb or 15 kb sizes. But if we have a higher percentage gel like 1%, 1.5%, the pore sizes will be relatively smaller, so it'll be better to separate out DNA molecules that are small, and then if the molecules are close to each other in their sizes, then you can easily separate them out. So higher percentage gel will give you better resolution. Resolution is the ability to separate out molecules that are very close in their sizes. Okay, so that's something you need to consider in your mind when you try to prepare your own gel. And then electrophoresis buffer, you probably made a TAE buffer, 10x TAE buffer in the solution prep lab, right? So that's what you're going to use. So either TAE, TBE, TPE, there are three types of buffers that you can use based on what acid you're going to use when you titrate the tris buffer in its pH, right? So in our lab, you're going to use TAE. And then your gel at the end of the electrophoresis run will be stained, right? So in our lab we use ethylene bromide, which is a very sensitive dye, but you need to be careful, as I said, because it is a strong mutagen and carcinogen, so you have to protect yourself from ethylene bromide. And then what's post-staining, pre-staining, there are two different types in the staining method, but we do post-staining method because ethylene bromide is a toxic substance. You don't want to contaminate a lot of, gel boxes or staining boxes and all those things. So post-staining will limit the contamination of ethylene bromide. So we use that method. And what is the size of the gel? I mean, the larger the size, you will have a better way of separating your DNA molecules. So that's something you need to keep in mind. And how much of DNA sample should you load? And yeah, so it's probably depending on the concentration of DNA. So you should have at least like 100 nanogram of DNA to be able to clearly visualize your DNA band on the gel. Okay, so it depends on the concentration. You can use whatever the volume you like. DNA sample preparation. So what kind of DNA are you visualizing or separating through the electrophoresis? Are you using a genomic DNA that's pretty large, like chromosome of DNA isolated from eukaryotic cell or prokaryotic cell? Okay, or are you just dealing with the plasmid DNA that are much smaller? Okay, it depends on DNA sample. Your preparation method will be different. And then, is your DNA cut with an enzyme? And the nuclease enzyme or not, and if it's cut, what is the predicted sizes of the DNA molecules, right? Depends on cut or uncut. If you're dealing with the plasmid DNA, we probably talked about three different conformations that they can have. So uncut plasmid DNA can have a super cold conformation, right, which is a pretty condensed size, and it may have a mixed circular DNA, which is a relaxed kind of a large conformation. And if it's cut, at least on the both strand, your DNA will be linearized, so they can come in different conformation. So based on the conformation, their migration pattern will be different. Understand that? And how close are the DNA molecules to each other? Of course, depends on the sizes. How sizes are similar or different. Okay, how much loading buffer should you use? So loading buffer in the electrophoresis box, okay, you don't want to use too much, you don't want to use too little. So it's right above the gel level is how much loading buffer you need to add. What size of standard marker should you use? So the size of a standard marker should be, you know, covering the size of your DNA samples, right? So your DNA sample size should be within the size marker range, of course. That's the answer. Gel loading condition, how much electrophoresis buffer is needed? I guess this question was how much loading buffer should be added. So depends on what loading buffer you have. Is it 10x buffer, 4x buffer? You have to calculate that mathematically. And then gel loading buffer, yeah, that the answer was, you have to fill the gel box right above the gel level. What's the voltage should be used? It's about 100 volt, and you run it until your loading die should be located on the right portion of the gel. How long the electrophoresis should be run? Again, you need to check the migration pattern of your loading die, so you can visualize it and you can determine when you need to stop the gel running. And make sure to make a correct connection with the wire, electric wires. Yes, that makes sense, right? So there are two different colors on the electric wires that will be connected to your gel or gel box. So one is black, the other is red. So black is negative and red is positive. So which one goes on top of the gel, close to the side of the gel where the wells with your samples are loaded? Should that be negative or positive? Of course, it should be close to the negative side, right? Because your electric current will run from negative to positive. So your sample should migrate from negative side to positive side. So that being said, then your wire with the black color should be placed on the top of the gel. Okay, so if you flip it around, your sample may run toward the opposite direction. It's going to run off from the gel. So that's a simple mistake you want to avoid, of course. Check the movement of the bromphene blue dye to make sure the right running direction. Yes, if you hook up the gel kind of opposite way, yeah, like I said, the DNA will run towards the opposite direction. You don't want that. After running is done, you're going to visualize it after staining with the ethylene bromide. Sometimes some labs may use methylene blue because ethylene bromide, there's always danger of contaminating, your containers or people. So methylene blue is an alternative staining solution, but the sensitivity is not that good. So you have to have enough DNA to be able to visualize your DNA band. So there are pros and cons on different staining solution, of course. How to document the result? Yeah, so normally you take a picture using a digital camera, and then you analyze the picture of your gel. How to interpret the result? Yeah, you need to see if the DNA molecules are separated, and then you can also estimate their sizes based on the standard marker bands. Okay, if you want, you can plot the standard marker, so the standard curve from the standard marker, and use that the mathematical equation that you can get from the standard curve, and then use that to estimate the size of your sample DNA. So yeah, those are important information. So for your lab exercise, you will be given with the plasmid DNA called POC19. Okay, so POC19 is a circular extrachromosomal DNA, pretty small, about 2686 base pair size. Okay, so that POC19 will be either uncut, untreated, or it'll be cut with various types of enzymes. So you're not going to do the enzyme digestion because it takes a long time. So our lab assistant Zach will prepare your samples for you. But keep that in mind, the size of the DNA library and the location of these enzymes where they're going to cut the POC19 depends on which enzymes cutting where. The resulted POC19 DNA band will have various conformation and the sizes. Okay, if it's cut once, it's going to give you that size, but if it's going to be cut twice somewhere, you may have two fragments with certain sizes. Okay, So sample preparation, you will be given it 4 samples. First sample is POC19 DNA uncut DNA. So it's going to be a maybe circular DNA. It may come in as a super cold conformation. Or it may come in as a Nick circular conformation. Okay, so that's what you predict to see. 2 possible conformation from uncut POC19 DNA, super cold or Nick circular, right? And then POC19 DNA will be cut with Hindi3 enzyme. So I don't know how many times Hindi3 would cut. So do you see Hindi3 from here? So it probably cuts somewhere in the multiple cloning sites. So I think, I'm not quite sure. So that's something you need to figure out. And the answer is right here. There's a Hindi3 cutting once in the POC19. Okay, so it's somewhere in the multiple cloning site. So this map is actually blown out a picture of that area, multiple cloning site. So there's that Hindi3 cutting site right there. Okay, so if it cuts once, you will see a linearized POC19 DNA, right? So 2686 base pair size you will expect to see from that DNA sample. And then POC19 is cut with PBU2. Okay, so I don't know how many times PB2 cut. You'll have to look at the map again, and based on how many times you cut, you will see, oh, it says a double cut. I guess it cuts twice. So you can try to find the PBU2 where it cuts. I don't know where. It's not showing up there easily, so you'll figure it out based on your result. Okay, and then the third sample will be the double cut. Okay, Hindi3 and PBU2 incubated with the POC19 together. So you can assume, since Hindi3 cuts once and PBU2 cuts twice, so by combining 2 enzymes, you may predict to see what 3 times POC19 will be cut by these 2 enzymes, right? Yes, that's what you expect to see. So, but I don't know what size fragments you will get. So you need to get the result from your experiment. Okay. So this is how your DNA molecules are treated. POC19, 3 microliter of DNA. I don't know how much DNA it is because we don't know the concentration. Hopefully Jack prepared enough DNA for you to be able to visualize them. And then this much different amount of water was added to each tube. And then KGB buffer, the potassium, phosphate and glycine buffer, base buffer. That's an enzyme buffer that activates the enzyme. First tube, no enzyme added, but second tube was added with Hindi3, 0.5 microliter. Third tube, PBU2 was added with a 0.5 microliter, and the last tube, 0.5 and 0.5 with change enzyme. Okay, so the total volume of each tube had about 15 microliter. Okay. And to that, of course, loading dye was added. So each group will make a gel. Of course, gel you need to prepare. So depends on what your preference is. Your group can decide if you want to make 0.5% or 1% or 1.5%. So at least I want you to have maybe 3 different gels from among different groups, so you can see the different separation pattern. Okay. So based on your calculation, so your gel has a size of 70 ml volume. Okay, then what is the amount of agarose powder you need to measure out? So 0.5% of 70 ml is how much? 1% of 70 ml of 70 ml is how much? 1.5% of 70 ml is how much? You figured it out, and then measure out the agarose powder and mix that with the TAE 1x TAE buffer, and then what? You have to heat it up to dissolve the agarose powder, right? And then you have to pour that heated up solution into a gel cast mold, and then let it sit so it'll be solidified. It takes about 15 to 20 minutes to get the gel set. Okay, and during the time you're going to separate, you're going to prepare DNA molecules with the loading dye. Okay, and then you will be given it a couple of size standard marker. One is lambda bacteriophage genomic DNA cut with Hindi3, and the next one is 5x174 bacteriophage DNA cut with Hay3. They have different size marker range. And then so you will add your sample, the 4 different samples with POC19 cut with different enzyme on cut, and then your size marker standards will be loaded to your gel, and then you'll run the gel about 1 hour, and then stain it, and then take a picture of that. So at the end, you'll get this information. Examine the size of the DNA band from the marker. So markers are used to so you can plot a standard curve, right? And then you can use one of the two markers, or you can combine the fragments from both markers to